Hello, friends. How are you all? I hope you all are doing well and I found you all in the good health. So, in my previous video, I had left you with some queries like why I'm using Arabic word in the case of fermentation, and the second one, I had assured you that I would discuss some interesting fact about the spirulina that are responsible for making the spirulina a widely used microorganism in the recipe production. So I would disclose all these facts in my video. So let's get started. This all these things we have discussed in my previous video. So from here we have to start. Now I would disclose the uh, first one query, that is why I have used the term aerobic in case of fermentation. Actually, what happened? The fermentation are of two types. That is the aerobic fermentation and the anaerobic fermentation. So, guys, what happened? Fermentation is mainly a metabolic process that is carried out in the absence of because of the three main reasons. First is the absence of the electron acceptor, that is the oxygen ultimately, and second, there will be malfunction, dysfunctioning or malfunctioning of enzyme related to the Krebs cycle, or absence of some enzyme that are related in the respiration process. So what happened? Fermentation is carried out in all of these cases. But what happened in aerobic fermentation is that in most of the SCP production, in case of SCP production, what happened? Aerobic fermentation is used. Why this is so? Because uh, I would discuss the advantage of it later. But first of all, we should know why aerobic ferment. What is the aerobic fermentation? So, it is a fermentation which carried out in the presence of oxygen. Fermentation is carried out. So, it is mainly used for utilization of the sugar. In this, what happened? A carbate effect is carried out, in which respiration is repressed by fermentation, except in condition of low sugar level. This is the well-known effect that is observed in the case of yeast. So the sugar level should be above the threshold sugar level to carry out the carbate effect. So aerobic fermentation is carried out because in SCP, we use aerobic fermentation mostly because it takes shorter time period in comparison to the anaerobic fermentation. And second is that it does not waste is it does not waste its energy unnecessarily in oxidizing the substrate to CO2. It just uses all the energy to produce biomass in more and more amount. So uh, here I'm clear why we are using the why aerobic fermentation is mostly preferred in the case of SCP production. So it is the list of the microorganism used for the SCP production. You can see it like in fungi, Aspergillus fumigatus, Aspergillus niger, Rhizopus cyclopium are used. In yeast, Saccharomyces cerevisiae, Candida tropicalis, and Candida utilis. Candida utilis is also called the torula yeast that is firstly used in SCP production in the World War first. I would discuss it later also. So, algae. Algae spirulina species, Chlorella parenidosa, Chondrus crispus are some of the algae that are used in SCP production. Pseudomonas fluorescens, Lactobacillus, Bacillus megaterium are some of the bacteria that are used in SCP production. So next, based on the nutrient requirements, bacterial population have been categorized into two types. That is the hydrocarbon utilizing bacteria and the hydrogen utilizing bacteria. So the hydrocarbon utilizing bacteria are further divided into the three types. That is the polyhydric alcohol utilizing bacteria, aliphatic hydrocarbon utilizing bacteria, and aromatic hydrocarbon utilizing bacteria. So, in polyhydric alcohol utilizing bacteria, uh, lactobacillus and streptococci are included. So, these utilize the simple sugar for its energy source, that is, the milk fermenting lactobacillus and streptococci utilize sugar lactose for its growth. Aliphatic hydrocarbon utilizing bacteria mainly use aliphatic hydrocarbon like methane, methanol as substrate for its cultivation, and they include methanomonas, methanooxidants, and methylococcus capsulates. Now next is the aromatic hydrocarbon utilizing bacteria utilize aromatic hydrocarbon, and they include Corynebacterium species and the Micrococcus species. Next is the hydrogen utilizing bacteria. In these bacteria, hydrogen serve as energy source for the strains, and the carbon dioxide serve as carbon source. Strains of Pseudomonas bacillus, Pseudomonas psychrophila, and etc. are widely used for production of SCP. The supplement of trace elements like potassium, magnesium, calcium, nickel, sulfur are needed for the better growth of these bacterial strains. Now, the production of bacterial biomass. As the upstream process of production include strains are isolated from the natural sources first of all, then they are standardized and pure culture are maintained an appropriate solid medium. So then the inoculum is prepared using appropriate broth. What I mean from appropriate broth culture is actually it should be free from contamination, and it should have the actively propagating biomass so that it can form the pure bacterial culture. And then this appropriate an inoculum is introduced into the bioreactor with optimizing growth parameter. Now the composition of production medium, supplement, and duration of cultivation may vary from strain to strain based on the cultural conditions. So the downstream processes for bacterial SCP production involve the centrifugation, flocculation, followed by killing and drying. The continuous production is carried out in case of bacterial biomass. Here you can see the systematic diagram of continuous production of SCP. Now, what you can see in this image is this is a motor with central shaft, and here is the impeller. This whole unit with the impellers, these are the impellers, and central shaft is called striers. Striers are used to agitate the medium so that the even distribution of the air could take place. Now, here is the water jacket, and here is the temperature controller. So the temperature controller detect the temperature. On the basis of that, for optimizing the temperature, the cold or hot water is passes through the water jacket time to time to control the temperature of the bioreactor in which our fermenters are growing. So here is the pH controller. So this pH controller timely detect the optimal level of the pH. This pH probe is inserted, that is the pH electrode. So with the addition of acid or basic ion, the optimization of the pH is carried out time to time. Here, here we can add the agriculture waste, food waste, and add any other substrate, and it is in the control, and we can add the substrate with continuously the substrate in the fresh medium to the bioreactor. Here is the gas controller. As we know, as the fermenter increases, increases in size, the necessary for the gases also increases. So necessity of gases also increases. For this, spargers. Spargers are used to introduce the small bubbles in the bioreactor, which a small bubble of gases in the bioreactor that are needed for the growth of bacteria. As we have discussed that aerobic respiration is carried out. So, uh, so the suitable amount or sufficient amount of oxygen should also be added continuously, should also be sparged with the help of spargers continuously to determine the good growth. Second, the production of SCP from algae. Now, the algal SCP has high content of protein with extra beta carotene, vitamin K, fatty acid, carbohydrate, and little quantity of vitamin C. The content of protein from algal SCP is approximately 5 per 100 gram of dry weight with low level of sulfur containing amino acids. A significant drawback in algal SCP is that it shows low digestibility and unpleasant taste. So the algal strain which are widely used for the production of SCP include Scindesmus, Chlorella, Chlamydomonas, Spirulina, Anabina, and Nostoc. So guys, in unlike the bacterial cultivation, algae are cultivated in the shallow ponds with continuous recirculation of the medium. As we know, sunlight is an important factor for the growth of the photosynthetic system of algae. So here you can see the image of the shallow pond. So shallow pond is filled with the medium containing ammonia and some necessary trace element with alkaline pH. The medium is circulated by a circulatory pump and continuous agitation is made by petal wheel roller with CO2 supplement. That petal wheel roller is attached somewhere here the bottom of the shallow pond and is continuously helpful in agitation of the medium. Okay, and the CO2 supplement are also important. So the unicellular algae are harvested by flocculation using aluminium sulfate and calcium hydroxide as effective flocculants, whereas the multicellular filamentous algae are harvested manually using nylon nets. Once harvested, the algae are dewatered and dried in open sand beds or using dryers. Now, the sufficient amount of algae produced can be harvested and can be used for the production of SCP. Now, spirulina. Now, here I would discuss something about spirulina. Recently, the production of SCP from spirulina has received great attention. Among the algal population, spirulina is widely used as a nutrient supplement since it possesses a broad range of nutrients. Other than the major protein and carbohydrates, it contains vitamin, mineral, and essential amino acids. Moreover, the cultivation of spirulina has several advantages. The first one is that the growth of spirulina will be high at alkaline pH and thus the contamination by other organisms is less possible. Second, since it has a gas vacuole, it floats over the medium, which facilitates its easy harvesting. Third, it has a thin cell wall, so it requires less heat during drying, and thus no need of spray drying like other algal strains. And the last one is that it shows high digestibility as it has less cell wall content and nucleic acid. Now here you can see the nutritional content of spirulina, which include vitamin, mineral, amino acid, fibers in this picture. So next we would discuss the production of spirulina from for SCP. Production of spirulina biomass. So the spirulina may be cultivated in semi-natural lake system or artificial ponds. However, Semi-natural lake system may yield less quality product due to the contamination by other organisms. On the other hand, an artificial pond system uses fresh water and provide necessary light, pH and temperature, and even a wastewater system can be set up. So in a wastewater system, the solid particles settle through the settle tanks and the effluent is allowed to flow into the artificial ponds. Spirulina is seeded over the effluent and allowed to grow. Then it is harvested manually using nets and dried with heat dryers. Next, production of SCP from fungi. So, fungal SCP is of much less importance because it has least quantity of amino acid methionine and high content of nucleic acid. However, the production has some significance because fungal strain can be grown in organic waste and some fungi can grow using cellulose as a carbon source, which is available in abundance as we know. Hence, cellulose is used as main substrate for the production of fungal SCP. So, fungal strains that grow over the cellulosic waste are Trichoderma species, Aspergillus species, Rhizopus species, and the Fusarium species. So, guys, yeast is another source of single cell protein and have been produced since a long time ago. In World War I, Torula yeast, that is the Candidus utilis, was produced in Germany and used in soups and sausages. Yeast has some advantages among other SCP sources such as food seasoning made from torula yeast. It is easy to harvest because of their size, that is, they are larger than the bacteria. They have the high level of malic acid content. It has high lysine content. It can grow at acidic pH. It has long history of traditional use. So there are few disadvantages that have to be taken as consideration, such as lower growth rate. It has lower growth rate compared to bacteria. It has lower protein content than bacteria. It also has lower methionine content than bacteria. So addition of methionine in the final product is carried out to solve this problem. Now the production is carried out by solid-state fermentation of semi-solid cellulosic waste and organic waste. Usually, the cellulosic material are treated with the alkali prior to inoculating the organism. This process help in the breakdown of the glucosidic bonds and facilitate the better fungal growth. The treated medium is then acidified to bring the pH to acidic condition. Further, the medium is amended with ammonium salts as nitrogen source and minerals. The pure fungal culture are seeded over the medium and the room temperature is applied. The recovery of the fungal mass is by decanting and filtration through porous plate filter. So these are the references which I have used, and thank you guys for listening me. And please like, subscribe my channel if you find it useful, and stay tuned with me to get updated about the videos related to the biotechnology. Thank you. Have a nice day.